Hello friends, welcome back to our channel Adami Education. We are back with the next part of cell organelles. Today we are going to begin with lysosomes. So lysosomes are also known as suicidal bags because they help in the digestion of intracellular materials which are no longer useful to the cell. Also it will help in the digestion of foreign substances or also the worn out organelles. It is actually budded out from the Golgi bodies. So you can say that endoplasmic reticulum will produce Golgi bodies and Golgi bodies will give rise to lysosomes at a later stage. And lysosomes, in order to dissolve or in order to digest so many different types of substances, it consists of 40 different types of enzymes which aid in its function. So as you can see in the diagram itself, the enzymes are located inside the lysosome and the lysosome is also covered with a membrane of its own. As you can see in the diagram out here, in the topmost diagram, the dark colored, black colored bodies, those are the lysosomes, and in the lower diagram, as you can see, the lysosomes are placed. They are not much numerous in number, or they are not very large also, but their main function is to get rid of all the worn out material in the cell. Now coming to centrosome. Centrosome is found only in animal cells. Its main function is in the help of cell division. Basically it will initiate the process of cell division. So as you can see, there are two structures in a centrosome. These two cylindrical structures are called centrioles. So you can say that a centrosome consists of two centrioles and outside there is a pericentriolar material. It is found only in animal cells, like I said you, and it initiates and regulates the process of cell division. It forms spindle fibers, thin hair-like fibers, which helps in pulling out the mitochondria during the process of cell division. In the next video, we are going to learn about the different stages of cell division in plant and animal cells. In that, you are going to have a much clearer view about the work of centrosome. Aster-like structures are formed. Basically, star-like structures are formed, which will actually produce the spindle fibers through which the chromosomes will separate during the process of cell division. Okay, as you can see in the diagram over here, the centrosome, it consists of two structures, the centrioles, and one more thing to be noted is the centrioles are always at right angles to one another. That's why in this diagram itself, as you can see, one looks like a cylinder, the other looks like a circle, because we are seeing it from a different view. And if you cut open a centriole, you are going to see such structures. You will be shocked to notice that if you cut open the flagella of a bacteria or of the algae Euglena, you are going to see the same structure. So you can say that centrioles and flagella, they have the same structure. Even cilia, the locomotory organs in case of unicellular organisms, they have exactly the same structure when you see a cut open view. Now coming to the next organelle, that is the plastid. Plastid is found only in plant cells, and the diagram over here, it is a chloroplast. As you can see, it has small coin-like structures and it also has an outer covering. Now let's see its functions. It is double membrane and it contains its own DNA. Like you remember in the first lecture, I told you that mitochondria and plastids are the only organelles which have their own DNA. That's why they are able to replicate very easily. They have disc-like structures, or you can say the coin-like structures. They are known as thylakoids, and these are the structures which actually contain the chlorophyll. There are three different types of plastids, and depending upon the color they give or the function they perform, they have been named. First of all is chloroplast, the most common one. They trap the solar energy for photosynthesis. Also, they will help in the process of photosynthesis. Now coming to chromoplast. Chromoplast will give different types of colors to the various plant parts. Even the fruits and the flowers gets its colors from chromoplast, and chromoplast has different types of pigments which give different colors. We are going to study about it in the next slide itself. Next, leucoplast is, you can say it is a colorless plastid. It will store starch. It will also give the whitish color like you see when you cut open a potato. It contains leucoplast in it. Like you can see, these are the microscopic images of a chloroplast. The structures that you see over here, these are the stacks which you just saw previously, the thylakoids. It looks very beautiful, to be honest. Next, coming to the various types of pigments which are present in chromoplast. Okay, xanthophyll will give yellow color. Like you can see, I have put some images out here. Next is beta carotene. Beta carotene gives orange color to the various parts of the plant or the fruits and the vegetables. Anthocyanin is responsible for giving blue and violet colors to brinjal, to blueberries, to various types of flowers, and lycopene gives red color to ripe tomato, etc. So these are the various pigments found in chromoplasts. Now coming to the most important part of the cell, which you can also say it is the brain of the cell, which performs all the necessary activities of the cells. It helps to regulate the activities of the cell. It provides all the necessary instructions to the cell organelles to perform their activities. It is the nucleus. So nucleus consists of an outer protective covering. Okay. Also in the outer protective covering, it has tiny holes through which materials can come in and go out. So the outer covering is called the nuclear envelope, and the small holes in it are known as the nuclear pores. Inside the nucleus, we have hair thin structures known as chromatin fibers, which will later condense to form chromosomes during the process of cell division. It also contains a fluid which is known as nucleoplasm, and it contains one or more round spherical objects which are known as nucleolus. The nucleolus also has a very important role to play. We are going to learn about it in the next slide. It is the largest cell organelle. It is very dense in nature and it is spherical in structure. It regulates all the functions of the cell. It has one or more round structures which are known as the nucleolus, and its main function is to produce ribosomes, and as you know, ribosomes help in the production of protein. So the nucleolus is responsible for the production of protein in the cell. It contains thread-like structures called chromatin, which contains the hereditary material, the DNA, using which we have attained the characteristics from our parents, and they had achieved it from their parents. So this cycle of attaining the characteristics from the parents to the offspring, that is done by the DNA, and this DNA is found in the chromatin fibers. The chromatin fibers generally during the resting phase of a cell, they do not appear very thick. They are just very thin, hair-like in nature, but when the process of cell division occurs, they become dense. They turn into the shape of X and Y, mainly X, and we are going to see about it in the next slide. Let's see. So this is the nucleus which we have, and inside this spherical structure is the nucleolus, and from nucleus only the endoplasmic reticulum is originating, like you can see in the figure. And this DNA, it is not just lying around here and there, it is found inside the chromatin fibers, and these are the structures of the chromatin fibers seen under a microscope. As you can see, it is so thin, hair-like strands, not very easy to understand. But when they become dense during the process of cell division, like you can see, they will form X-like structures. Now this Y structure is found only in males, because as you know, XX is for a female, XY is for a male. Males will have the last sex chromosome as Y, and females will have it as X. So this this chromosome combination is that of a male, and this is available to be seen under the microscope only during the process of cell division, not before that. These chromosomes are of different shapes. There are different types of chromosomes. We will have a different video on that, or maybe I will put it as a shorts. So definitely go there and watch it. You will be astounded by the fact that there are so many different types of chromosomes available in our body cells. Next, coming to the last organelle, that is the vacuole. Vacuole is generally found in plant cells. Quite a large vacuole is found in plant cells. Sometimes they can also be found in animal cells, but they are quite inconspicuous and small. So vacuole consists of water or other food materials, gas, or generally in case of plants it contains cell sap. It is found generally in plant cell, like I already said to you. It consists of an outer covering which is known as the tonoplast, quite important, and it gives turgidity to the cell. The vacuole is responsible for storing water. So when the plant cells are turgid, that means they're quite inflated. The vacuole contains enough water. Now if you put the plant cell in a salt or in a sugar solution, the water will be removed from the vacuoles, and the plant cell will become flaccid, which ultimately kills the plant. We are going to see this in the next video as well. What is turgid, what is flaccid, what I am talking about, we are going to see all of it. Please wait for the next video to learn about it. So basically, the main function of vacuole is to provide turgidity to the cell, also to store the necessary water or gas or other materials in it. So this is the real life picture of a vacuole. As you can see, it is quite large, and these are both the cells of plants. Now coming to what are the differences between plant and animal cells. Okay, before that let me tell you, other than this, in the cytoplasm itself you can find many different granules of fat, protein, etc., just materials floating here and there. So those are not actually organelles, so I did not include it in this, but they are also found in a cell, generally in an animal cell, because they don't have vacuoles to store those, so they are just found floating in the cytoplasm itself. Now coming to the differences, in case of plant cell, a cell wall is present which is made out of cellulose, and in animal cell there is no cell wall. In plant cells there is no centrosome, like I already said to you above. In animal cells the centrosome is present. In plant cells the vacuoles are quite prominent and large, but in animal cells the vacuoles, if any, are very inconspicuous and temporary, only necessary when requirement is there, otherwise it is not present. Plant cells contain plastids, like we discussed. Animal cells do not have plastids. In plant cells we will see the cytoplasm is not very dense and granular, like I just said you, but in animal cells there are a lot of granules which are found in the cytoplasm. Plant cells are generally larger in size, animal cells are smaller in size comparatively. In plant cells the cytoplasm is not much present because the vacuole occupies the most of the space, but in case of animal cells the cytoplasm is quite occupying a larger space inside, because cytoplasm performs almost all the necessary functions along with the intervention of the nucleus. So that's all for today. I guess you have understood the lecture. This is one of my favorite chapters, and it made me very excited to share it with you. Hope you enjoyed it as much as I did. Keep waiting for the next video. We are going to learn about the things which I said you previously. So bye everybody, keep studying, and in case of any doubts or any questions, please feel free to comment in the comment section provided below the video. Bye everybody, have a great day ahead.